So, I think I'm online again. Hi, hello, and welcome again. Microbehunter here, and welcome to another microscopy live stream. Like all time, every time I'm just trying to get everything running and going. Can see myself because I also check. Yes, I can also hear myself. That's good news because if you've been present last week, you know that I had a little bit of a panic because one of my cameras started to fail. I think I've got everything sorted out now, and yeah, I basically upgraded my computer a little bit, and I hope that the connection is now a little bit more stable here. Yeah, you can see there is a little bit of chaos right now on my desktop here. I've got a whole bunch of stuff standing around here, right? Today I would like to really try out different mounting media. And also, especially if you are from a German-speaking country, look, this is a new mounting medium that I found. It's glue, right? And I would like to try this out as well. This is a water-based glue. So specifically, I would like to try to mount some so-called diatomaceous earth, which are diatoms, fossilized diatoms. And I would like to then check how well this actually worked. And because I know that every week people also want to see some moving things and some microorganisms that are actually living. I've of course also prepared then later on towards the end a small water sample which we can just observe just for the fun of it. But indeed I do want to test this out. And for those of you who know this format, this is a little bit non-scripted and unprepared. So indeed, I've not the things that I'm showing you here live right now, I have not tried out myself. And this is, or at least not in that detail. And for this reason, this is a little bit a very authentic thing here. As always, I would like to say hello all around the world, to Vietnam, to Belgium, to the Netherlands, okay, to the United States, to the Netherlands, to Germany, Florida. Okay, I'm just reading this here. UK, Texas, USA, Athens in Greece. Okay, PVA glue. I don't know if this is PVA glue. Honestly, I don't know what it includes. Okay, it just says it's basically it does not contain any solvent. Of course, it contains water as a solvent. Yeah, and if you have any questions, related to the topic directly, then of course I will try to answer it directly. Otherwise, all off-topic questions, please do post them as well. Always write down at Oliver or at Microbehunter, so it's easy for me to find because I get many hundred comments, which is really great. But sometimes it can be difficult while I talk to actually find the comments that are for me. And and there's a question from Marcel. How did you solve the USB problem from last week? I got from Jesse the recommendation to, and I didn't even actually have to admit I didn't know that this actually exists. I have 6 cameras connected to the computer, and of course the USB system, those USB ports were overloaded. Not only from a data connection, but also from the electricity, from the power. And I bought myself a second USB card, one where you actually get more power. You have to also connect the power. And ever since then, it works more stably. No guarantee, but I did not have any issues. I didn't have any issues, but one always has to be a little bit careful. I'm glad that essentially I got that problem, at least for now, resolved. Okay. So hi, Oliver from England, from Romania, from Austria. Yes, also hello from Austria, from Upper Austria in my case. And there is already a comment here. I use Plurax for Diatom mounts. I don't have that. Okay. Do infinite and finite microscope share the same eyepieces? I can talk about that later. And very briefly, it depends, but generally it is possible. Okay. And So hello from Hungary. Great. So what I will do is the following. I'm just going to get started and give you a little bit of an overview of what I want to do today, actually. Okay. So it's, as always, as I already mentioned, it's things like these are also always kind of experimental and ad hoc. So we'll see how well this works out. But I have a whole bunch of mounting media, some of which you already know. UPRO mounting medium that's expired 2013. It still works, right? It's usually used for insects. This one here is you already know that Elmer's PVA glue, a water-based glue. This here is nitrocellulose. It's a nail polish. right? Usually it contains nitrocellulose, has a disadvantage that it tends to shrink a lot. Yeah, so we are aware of that. Over here, this mounting medium I cannot use, unfortunately, anymore because it started to become too thick, so it doesn't flow anymore. So I have to add a solvent to that to make it liquid again. UKIT is a resin-based mounting medium, and actually you should actually not use it at home. You should actually have a proper ventilation, some kind of a fume hood or so. It smells nice. That's not the problem, but it's not apparently not healthy to inhale. Okay, then I also would like to try this new glue, of course, here that I have not tried yet. I suppose it's that's also PVA containing, I suppose. And then look what I'm gonna also try, and this is also gonna be the first time that I tried. I'm gonna try to use pure glycerin, okay, as a mounting medium. And I would like to try to mount it using those slides that I prepared here. Okay, so I made a ring using nail polish to prevent the glycerol to actually come out here. So I will try if this actually works. Okay, so this is something that I would like to try. And if you have any questions or any comments, then do please yeah, post them here every now and then. I will actually try to read the comments here. Yeah, yeah. Okay, so that's the thing here. So, and I don't know, I'm just going to get started here by taking out some of this diatomaceous earth. And I've made some videos already about that. And every now and then I keep on coming back. I ordered this from Amazon. Yeah, this is how it looks like. Fossil shell flour. Why in the world do they even sell this stuff here? I mean, yeah. And yeah, apparently some people like to eat this. Okay, I don't know what language it is. It could be Polish or something. I don't know. In any case, I'm not going to eat this stuff, obviously. However, it does look quite nice under the microscope. And this is called diatomaceous earth, and it has a variety of different uses. So it can be used as a filter material. Yeah, you can mix it yeah, with nitroglycerin to make, yes, dynamite. They used to make dynamite using this. I've heard that they used to put it also in some toothpaste as a kind of as abrasive. Apparently it cleans the teeth better. So it has a variety of also industrial uses as well. Yeah, these are basically fossilized diatoms, and they look okay, they look nice. And the reason why I want to use those and not just natural diatoms, which I also have in my water samples, is because it's pure silica. This basically means that if I want to test which mounting medium actually works best, then I think this gives me a more controlled you know, sample. Why the different mounting media? I also need to explain this because refractive index is important. Okay, so I'm just going to take this little thing here. I'm going to take a little bit out of this because this is way too much. And the different mounting media have a different refractive index. I think this is more than enough already, right? Normally you want to have a, and this is now where I'm going to just talk a little bit about some theory here. Generally, if you want to observe, and I'll be putting this under my phase contrast, objective, you want to have a refractive index which is different from the refractive index of the diatoms. Otherwise, you're not going to see it because they're transparent. It's made of glass, so to say. Silica is like glass. It's transparent, and if they're now surrounded by a mounting medium that has the same refractive index, basically you're not able to see anything, right? So yeah, it's so you want to have a mounting medium with a very different refractive index, and that's a little bit a test that I'm doing right now. I just want to see how this actually looks like under the microscope, and I'm well aware that it takes some time for the mounting medium also to dry and to become solid, and for this reason. We're probably going to look at the slides next week again. Okay, so just saying. Yeah. So what I'm going to do now is I'm quickly going to scan again the comments here. Okay, hello from Armenia, from Netherlands. Okay, I skip over. I'm just looking at okay. Yeah, somebody just returned from the hospital just to see my live stream. Okay, yeah. I've just started trialing with artist watercolor. Okay, yeah. So I'm just going to skip. Yeah, you have to be very careful not to breathe the dust from highly damaging to the lungs. Yes, it can. It's called silicosis or something like that. Yeah, not healthy. Yeah, so I'm well aware of that, and yeah, you have to kind of be careful that this yeah that you don't inhale those silica things. Yeah, it's used as an insect repellent. Yeah, so is a diatom a plant species or an organism? Well, plants are organisms. Diatoms are related to the protists, they're single-celled. I mean, you can say that they're also related to the plants because they do photosynthesize. They're category on their own, really. Yeah. Yeah. How about staining these diatoms? I'm not so sure whether staining is actually going to work well because you'd have to stain silica, and I'm not so sure whether it actually stains well or not. Yeah. So what I'm going to just try to do now is I'm just going to try the most basic one, and I'm just going to try it in pure water. Okay. And in order to do that, of course, I need some water. I'm just going to put some water in here. And we're just going to use this as a reference. Okay, yeah, simply to see how this looks like. And yeah, you already know these things. Yeah. You want to use very little of this substance as well. You know what I could try? Maybe I'm going to try again my toothpick here. You can see that my coffee warmer is also here because I'm going to also use yeah, the glycerin gelatin later on. So you suspend a little bit of this on here, and then you need, of course, a cover glass. And one of the things that kind of annoys me with these cover glasses, as you already know, is that they are a little bit, it seems to be okay. Otherwise they're kind of dirty. And here we go. Yeah, it looks okay. And let's put it under the microscope. And now the hour of truth. Does it work? Does it work? Does it work? Does it work? It works. I'm so happy that the thing works now more stably than before. Of course, it's way too bright. Yeah. It's completely out of focus. Open the Köhler. This is how it looks like. Air bubbles, of course. Okay. And let's zoom in. 20. Let's stay with 40 times because I'll be also using phase contrast at 40. Okay, and here these are marine diatoms. I'm just going to use bright field now so that you that's bright field. You have a better comparison because most of your microscopes that you have is probably also bright field microscopes. Let me put the trash can away. My desk is very cluttered, very, very cluttered. Yeah, so these are basically those diatom shells that you have here. If you just want, let's just go up to 60 times so that you see them a little bit better. Look, actually, when I focus, yeah, here we go. Yeah, that's 60 times. Yeah, and that's 40 times. And but I'll be looking at them today mostly with phase contrast, and the reason is because differences in refractive index can best be seen in phase contrast. And so what we see here is that it looks quite bright on. Yeah, they look quite bright. Yeah. So let me open this here. So that's basically how they look like, right? I know they don't look. Yeah, I mean, beauty is in the eye of the beholder. But considering the fact that these are fossils, really, yeah, it's also quite remarkable. And basically, what I will be using is I want to now make different slides with different mounting media, and I would like to compare it, right? How's the contrast change, if at all, right? And because you should be using specialized diatom mounting media, and there's several of them, and because the mounting medium should have a high refractive index, so that you're actually able to see it. Because if it has the same refractive index, you wouldn't be able to see anything. Yeah. So this is a little bit the idea here, and so what I'll be doing therefore is I'll be just making a whole bunch of microscope slides now together with you. Okay. And if you have any questions or comments or whatever, please do mention them. And yeah. And then what I said is that maybe towards the end, I'm going to abruptly switch topics, and I'll be also putting just some water samples under the microscope just for the fun of it. For those of you who want to see some moving things as well. Okay, but for the sake of the learning aspect, also I like to do these experiments here. Okay, so let's go back here again. Okay, and let's simply get started. Okay, and what I'll be doing is I'll be using those slides here. They have basically a matte area here where I can actually write on here. The water one is now in here, and I'm just gonna, just because it's here, I'm just going to use now nail polish. So I have to label the slides first. I'm just gonna write it maybe like this: nail polish. So later on I can always wipe it away with alcohol and then put a proper slide label on top. But this is an experimental, and therefore, yeah. So, question is how I'm gonna do that. If I put the diatoms on here first, and then then I might transfer some of them into the nail polish. So I'm just going to do it like this. I'm going to do it the easy way. Just a little drop. Okay. And I'm going to pick up, I'm going to always use a fresh, the fresh side, and we just go like this, and we're going to give it a try. Properly suspend it, and then the cover glass, I just throw it away directly into the trash can, and cover glass goes on top. Yeah, it's highly viscous. We press down a little bit. Yeah, and just for your information, previous experience has shown. Let me turn off the lamp here. Yeah, so there is no reflection. Previous experience has shown that indeed the nail polish does tend to shrink. Oh my gosh! Thank you very much for the donation. Okay, thank you very much. Okay, so what I would like to do now is, let's have a look. Let's have a look at this slide here. If nail polish shrinks, I mean, first of all, before I put it under the microscope, nail polish has been used as a mounting medium. As a matter of fact, someone I know who's been working with so-called testate amoebae. So basically, these are the tests are the so-called the shells of amoeba, and he's been isolating them from sand samples for his PhD work. And he told me that he used standardized in the lab. They used nail polish. It's nitrocellulose. Apparently, it worked quite well. I found that it tends to shrink a lot. My recommendation is that if it shrinks too much, then allow it to dry first, and maybe it contains too much solvent, make it a little bit more viscous, thicker, okay? So, and another thing I need to say, of course, I'm very careful now that there is no mounting medium spilling out from beneath the cover glass, because I do not want to get any of this mounting medium on my objective, because I'm working right now with a 40 times objective, and this means the working distance between the objective and the cover glass is very small. So you got to be super careful that you don't get any of this stuff on your objective. If you get some water on your objective, you can always wipe it off, but this one is going to be more difficult, okay? Just as a general warning, because I'm, as you can guess, I'm talking a little bit from experience, okay? So that is, I'm going to put in the slide now, and let's have a look, okay? So this one is now the one with clear nail polish. And I have to find it first. Let's see. A lot there. They're the completely wrong area. Here we go. Okay. And you know what? I'm gonna put the water slide next to it. Yeah. So that we can quickly move over to the water, so that we can compare. Okay. So that is now using nail polish. And now this here is using water. Just ignore the density, okay? Ignore the density, of the diatoms. Of course, the more diatoms, I got more here, but over here, this is the one, with nail polish, and you see that the contrast is quite a bit lower. Maybe I should have taken it. I should have maybe taken slightly more. But if you see them, those little square box-like structures, Here are slightly more. Yep, they don't look quite as bright. I'm not so happy with the place here. Here, there's slightly more here. Yeah. Yeah, you see that they don't shine to light up quite as much, which is nothing unusual because depending on the difference in refractive index with the surrounding, this is will determine how well you're able to see them. And if the refractive index is too similar to the diatoms, then you would not be able to see anything at all. Okay, because then the light is not phase shifted, as it passes through the specimens. Yeah. So just wanted to okay. But again, maybe the whole thing changes. The refractive index changes as it dries. Okay. So that's nail polish. So we will have a look at this later on again. And then we're going to also see how much the specimen has actually shrunk. Okay, the next time I'll take a little bit slightly more. Okay, so then what I'm going to do next is I'm going to try then Elmer's PVA glue. So that's a water-based mounting medium. So, and then I need to change over always to the desk again. Okay, so this is Elmer's PVA glue. I forgot where I put the bottle. Just a second. Here it is. Yeah, you already. Just for those of you who are maybe new to this, I found this online. You got to be careful because many of the glues are white glue, right? And sometimes, it can be kind of difficult. You want to have the clear one, right? And it's water-based. It's child-friendly. It's non-poisonous. And PVA actually is an official mounting medium. But this, because if you want to get it laboratory grade, it's going to be expensive and so on. So for hobby purposes, stuff like this also works or might work, right? We're not doing this for how do you say standardized scientific research, but we're doing this actually just for the fun of it. So and what I've done is I've taken this PVA glue and I have basically added a little bit of water to it to make it a little bit thinner. Okay, and we do the same thing here. Yeah. Okay, bubbles are sometimes difficult to avoid. So, and let's take a little bit more of this. Here, let's add a little bit more here to make it a little denser. I think this should be fine. You want to mix it a little bit to break apart any clusters. When you add a cover glass, very often clusters will break apart anyway as it's pressed flat. But so, and we drop another cover glass on here. And don't forget to label it, because otherwise I will really not forget. I will really forget Elmer's PVA. PVA. So, it's pressed a little bit. And again, there is nothing spilled out, which is good. And let's give it a try again. Oh yeah, I need to change scope. So, you see the glue is still spreading. Oh yeah. Yeah, here again, because it's maybe because water-based, the contrast again is a little bit higher. Yeah, and it's a little bit like honey, and it's floating as the glue kind of evens out, and the tensions inside the glue and the cover glass kind of even out. That's why it spreads, and it's going to. That's why it moves, and it should actually stop moving. Yeah. Yeah. So, and while it does this, I'll quickly. Do you still use a large syringe to remove air bubbles? Actually, I promised myself that I will answer these questions separately, but just a second. Look at this. I've been actually experimenting with this. You can do that. It's a highly improvised system. And as you're watching it, what you can do is you can put in a small sample in here. For example, today, originally before I wanted to actually do the whole thing here with the diatoms, I was actually thinking about making very thin cross sections of wood. Here's a piece of wood, and then removing the air from the air spaces by using a vacuum. But it's, yeah, and it does work a little bit, but then I came up with the idea of using those diatoms. See, there's a gigantic air bubble now approaching. Yeah, and what you do is I just basically you put the sample in here and with a little bit of water, and then you just hold it closed here, and then you just very tightly, and you just pull, and then this basically will pull out the air from any air spaces. Yeah, so yeah. I've gotten some nasty ideas. How do actually microorganisms react to high pressure or low pressure? That would be also. Okay, I'm a mad scientist. Okay, yeah. So this is let me go down with the magnification. Yeah, that's this air bubble, right? Yeah. So this is a phase contrast right now, as I mentioned. But look, if I switch over from phase contrast to the 4 times non, then you get dark field. Yeah. So just for your So you see. Again, what do we learn from this? Let's compare this PVA glue with the nail polish. Okay. Ignoring the fact that actually I didn't. Maybe the density of those is. These are all contaminants here. That's Now you see the diatoms. Yeah. Here they are. I should have probably taken a little bit more. Yeah. So, and this here is, again, you see the focus is different as well. Yeah. And that's, again, the PVA glue. Okay. That's actually quite a nice one here. I kind of like this. You know what? I'm going to now do the following just for the fun of it. Let's just have a look at it. I'm using DIC 60 times. And... yeah, 60 times DIC. Let's give it some color. Yeah. All right. So that's basically, here they are. And now let's have a look at the nail polish. See, do you see all of these things here on the surface? That's basically stuff that's that was not on the microscope slide, but actually on the cover glass. Yeah, it's kind of. Here we go again. You see that, and in bright field, so it looks like in bright field, the nail polish, and that's the Elmer's PVA glue. Do you actually see that when I focus back and forth, how this actually pushes? The reason is because, it was touching the slide. Yeah, see, that is actually, I think the Elmer's PVA glue, as you see them actually a little bit better even. And you know what? See, there are in the nail. You know what I'm gonna do now? I'm not happy. I'm not happy with this at all. I'm going to make the nail polish again, but I'm going to use a I'm going to use a higher density of cells, not of cells, of diatom shells. Yeah, so let's do this again. Where is the nail polish? Okay, and let's use more. Again, I flip the toothpick. Here, that's a lot. Always make sure that it's properly submerged so that any air, goes out. But I find those diatom shells very water soluble in any case. Now this was a lot, okay? So definitely enough. So let's give this one here a try. Because, okay. Here we go. And we push down again to really make sure that everything is very thin. This is not a lot. So always label it nail polish. So let's take out the old nail polish slide and add the one now where the concentration is higher. Scope view. Here we go. Too bright. Ah, much better. Okay. So, where is the phase contrast? And we open the condenser. Okay, yeah. So, very nice. So you see how this looks like here? Okay. It's fairly dark still. The individual diatoms are not so well visible. And the PVA glue is here. Yeah. So refractive index doesn't matter. Okay. So let's move on. Let's try different. Let's try different mounting medium. Yeah, so the diatoms reproduction, indeed, they basically become smaller and smaller, but then basically what they will have is they will regenerate this. So there is then a break in this system, and then they will divide in a different way. I read somewhere, and then this will basically restore the original size again. Yeah. Did you ever try blue nail polish? Would it color the background like the aceton oblique light? I've never, honestly, I think blue nail polish would be a little bit too dark. We can try this as well, but yeah. So let's move on again on the how do you call this, the desk view, and let's try the next one. Okay, so this I've tried now. Yeah. Then the clear nail polish, I've tried the Elmer's PVA glue. Let's move them away. Where did I put it? Funny, I misplaced the bottle. Over there it is, fine. So, I'm going to try now this glue here. If you're from a German-speaking country, you're very well aware of this. Yeah, it's, yeah. And this one over here, normally the regular glue contains a solvent like acetone, but this one over here says that, yeah, without solvent. Obviously there is a solvent in here which is called water, right? So I suppose that, yeah, it's similar to PVA glue, I guess, right? So I've never tried this, and I've not diluted this. So this is probably gonna be a little bit thick, okay? So normally you would like to thin it down a little bit, but I'm going to just do it like this. It's very thick. Yeah. And we try it again. Again, we just ignore the air bubbles. Okay, I think that's also enough now. Okay. And when it starts to absorb the water, then you will see that the whole thing, the diatoms become more and more transparent, because they have a similar refractive index. So, yeah, let's again trash this. Cover glass goes on top. Aha, who is also, yeah, in England. So, let's give this one here a try. I think that's a lot what I've got here. So let's give it a try. Okay. I need to go to the scope view. Yep, here it is. Yeah. Yeah, it's also not quite as bright as Elmer's PVA glue. It's interesting. Yeah. I mean, it is, they are visible, yeah, but not quite as bright as the other one. It would be kind of nice to have some kind of a system to actually measure the refractive index somehow. Yeah. That would be kind of, but I wouldn't know how to do that. Yeah, yeah. But you see that it's fairly dark. Okay, and again some yeah. Where they're clustered together, you see that there's still some air bubbles. The view in bright field must be better than in phase contrast. It might be, but in phase contrast you actually see it better, the differences in refractive index. So let's switch over to bright field, why not? And I close down, yeah. And this is how they look like here. Bubble. And later on I'm going to show you some live diatoms. So next one, I want to try to make, let's do the following. What time is it? I always have a little bit of a time issue as well. I'm going to now, where's the desk view? I would like to try now something a little bit, I have to, just a second, I have to label this first here. Yoo-hoo. So, and the next thing that I want to try is the following, and I don't know if this is actually going to work out or not. This is a little bit risky. It is possible to make slides using pure glycerol. But the question is, how do you make this permanent, right? And you know what, I'm just going to make a normal slide first, and then later I can try to use this. I made, using my slide ringing table and some nail polish, I made this ring here to prevent the glycerin from actually spreading out on the side, so that they can actually seal it off from the side, right? This is kind of my intention, but just for the first trial, we might simply make a regular one. We just apply a small drop of the glycerin or glycerol, same thing, on here. It's a little bit like an oily substance, but it's actually water soluble. I think a small drop is enough. I trash the rest. Always close it off immediately so that you doesn't tip over. And we now do the following again. And if this kind of works, I would like to try the other one as well. So, we mix it properly. Let's try, see how pure glycerin works. Yeah, it spreads much nicer and much thinner, of course, because it's so liquid. And again, scope view. Okay, that's bright field again. Okay, also nice. Let's see. Let's go back to phase. And then I might want to do a side-by-side comparison. Also, look, also very bright. Okay. Yeah, so it's again a little bit dense, I would say, but also very nicely visible. Why not go up here 60 times? Yeah. It's again bright field at 60 times. So there is a question about UV resin mount. I've not tried that yet. I did buy myself a couple of months ago. There is this nail polish that cures in UV light. But it was too thick. It was like a gel. It was not liquid enough, so I had problems actually mixing in the diatoms. Yeah. So that is basically so glycerin works. Now I would like to that's going to be now the experimental part. I would like to now try to try to do it like, let me remove this here again. I would like to now, put the glycerol in here. and then let's see if I'm able to actually put a cover glass on top here and also able to seal it off. I don't know. This is going to be somewhat of a difficult thing. I don't know, but let's give it a try. Yeah, it's probably not going to work well because I don't want to work now with a slide ringing table as such. And we need that's already a lot. That's already a lot. Maybe it's too much. I don't know. It could be. Yeah. Let's give it a try. So that's enough. So, and now we try to put on a round cover glass. Here is a round cover glass. I hope I got the diameter correct. Okay. You see that it acts like a spacer. And now, what I would like to do, and this is just for, how do you say, for demonstration purposes. Normally you would probably do this a little bit differently. Normally you would probably apply first another ring, but I'm going to just try to do the following now. I'm going to now try to use this nail polish here. And I would like to, yes, it is on here. And this is going to be a little bit messy, and maybe it's not going to look quite as nice anymore. Yeah, I can always scratch that off later on. But I would like to now it is going to be a little, and hope that capillary action is able to do the rest. Normally it should get drawn in a little bit. I don't know. I've done this in the past. It usually got drawn in a little bit, and maybe it's not liquid enough. Yeah, and this is how I was able to seal off the cover glass. Whatever. Or what you do is you try to rotate it again, and you try to apply another layer, right? Or you try to apply the cover glass when the first layer, the ring, is not entirely dry yet. Okay, so I just wanted to, that's a little bit some of the things that I have been playing around with. Yeah, and then what you have to do is you have to then try to scratch off the rest. Yeah, so this is going to be a little bit dangerous now because it's fairly high. So I'm only going to use a low power objective where there's enough distance. And yeah, this is how it kind of looks like. Yeah, this is a so-called a semi-permanent mount. Yeah. So I'm going to. What about trying a hydrophobic mounting medium? That's the next thing that I would like to do, and that's Uparol is a hydrophobic one. Actually, nail polish as well. Yeah. Yeah. So that is also a recommendation here. I'm just going to go through here. You could take a raw photo on the DSLR and then compare the histograms for a low-level RI test. That would be a possibility. That's actually also one of the things that I want to try when everything's properly dried. That simply take using the same settings, manual adjustments, and then I can actually do a side-by-side comparison. Yeah? And the more different, or let's put it this way, the more different the contrast is, the more different the refractive index. Yeah? Why is using glycerol risky? Let's put it this way: it's not risky from a safety aspect, but it's risky because it does not solidify, it does not turn solid. Yeah, and this means that if the glycerol actually touches now the side, then it might actually, due to capillary action, prevent the cover glass from sticking. Yeah. So this is actually something that I've read also somewhere once. As soon as something is covered with glycerol, it's nothing's going to stick on it anymore. So you have to be careful that the glycerol essentially does not contaminate those areas where you would like the cover glass to make contact. Yeah. So this is the reason why this is somewhat of a delicate issue. Okay. So, but it's kind of glycerol or glycerine. So the next thing that I would like to try is the following. You know this already, right? And this is the mounting medium which you have to heat up. A couple of weeks ago I already was staining some pollen using this mounting medium here. It's solid and you have to warm it up. So basically you have to put it on here, on a coffee warmer. But it's dangerous because you're not allowed to make it too hot, right? And if I put it to 40 degrees or so, it takes maybe over an hour, 1.5 hours to actually melt. So I've read somewhere in the book there is a second possibility how you can actually do that. And that means you do the following. And I've also not tried that. I remove this. And that is also a little bit, it's already pretty warm. Okay, what you do is you try to melt it directly on here. But this is definitely warmer than 40 degrees. Okay, so one way of how you could actually do this to prevent it from becoming too warm, because once glycerin has overheated, it won't solidify anymore. The proteins, gelatin is made of proteins. Yeah, maybe like this. Yeah, and they might denature. Yeah, I have to remove this here. This is how it looks like. And in the book, what they recommended is that you melt it directly, and I have not tried this. I need another spatula. They recommended that you essentially don't melt the whole thing in the jar, but that you actually go in here and you take out a little piece of this directly. This is pretty sticky. I don't know, this is a little bit, you shouldn't be touching this. And then you try to melt it directly on the slide. Yeah, and I think it's already starting to slowly melt. Yep, see? Becomes liquid. So that's actually, I think this is actually way faster than just waiting for over an hour for the whole container to become liquid. Yeah, you know what I'm gonna do now? I'm just going to go in here. Now that it's sticky, it might actually. So, same story as before. Now this glycerin gelatin actually contains a stain which is used for staining pollen. So I doubt that this actually also will be able to stain the silica shells here. Frustrules, they're called. So, and I'm just going to use a square cover glass again. Normally you're supposed to seal it off later on, so I'm just going to put this one here on here. And the whole thing goes again under the microscope. So where is the scope view? Yep, here we go. Oh yeah, it's again DIC. Let's go again. Phase contrast. Yeah. Also not quite as bright as the other one. Yeah, so basically, yeah, glycerin gelatin also is fairly dark. So this means that the refractive index is very similar to the diatoms. Huh? Yeah, but that's how we learn. Yeah? Kind of illustrates that yeah, the correct choice of mounting medium can indeed make a big difference in appearance. Yeah. So I just have a look here. So request for hydrophobic mounting medium. Yeah. So basically this is yeah, the glycerin gelatin, and let's try to close this here. Yeah, otherwise here it looks quite okay. Yeah, yeah, it looks actually quite. It's a bit messy. Yeah, that's again DIC. If you see it with a colored background. The refractive index will most likely change, yeah, because well, maybe not so much with the glycerin gelatin, because there's not so much evaporation happening; it just cools down. But for the others, where the solvent evaporates, it will most likely actually change the refractive index. So that means we have to check this again next week. Yeah? So let me quickly go back to the camera, the desk view here, and this one is, I have to label this one here, is glycerin gelatin. Glycerol jelly. Okay. And you see it's slightly pinkish because of the stain that was added. So, and then I would like to try, last but not least, no, I've got another one that I would like to show you. Last but not least, the U-Pearl mounting medium, which is actually quite common to be used for insects, because it has this natural clearing action. Yeah. And here again, we just do the following. It has a slightly yellowish color. This one can actually be diluted also with alcohol. You see, it's already a little bit solid. Yeah, and we now try. Okay. Ah, yeah. I'm just reading. Every time when I'm pausing, I'm occasionally reading the comments. Okay. That is a hydrophobic mounting medium. So, and usually it takes a fairly long time for this mounting medium to actually dry. So usually around about a month or 6 weeks. Depends a little bit also on the temperature. Yeah. So, but you got to be patient, and during that time you should always store it horizontally. Yeah. So we have again here, carefully repress. Yeah. And we have another little check here under the microscope. And yep, this is how it looks like. Again, I will try to do that with... Ah yeah, here they do appear to be again slightly brighter than in the previous one. Right. So maybe I'm just going to make some kind of a ranking, yeah, which ones actually look the brightest. Yeah. Yeah, so they here it's again a little bit, but not quite as bright as the first one. Yeah, so we have again bright field. Yeah, this is how it looks like. So that's U-Pearl mounting medium. Yeah. At these low magnifications, I think everything looks quite okay. Let's try the PVA. Let's try to see if we see a difference here and here. So that was the U-Pearl, and that's the, oh, there's still some clusters here. So that is the Elmer's PVA glue. And that is the U-Pearl one. Okay. Yeah, this can be a little bit misleading because yeah, of the different densities yeah, of the shells. Yeah, I'd probably have to zoom in into one specific one and then make a direct comparison. So this one here was U-Pearl. I have to also label that. Desk view. U-Pearl. Okay. So the common question is, what would it look like if one of the substances had a lower refractive index? Would that be darker? Yeah, yes. Generally, it is like this. You should be using for diatoms, there is special mounting media that have a very high refractive index. And the higher the refractive index is, the more different the refractive index from those from the diatoms, and then you should be able to see them better. Right? Because if it has exactly the same refractive index, the light is just going to pass through unhindered, and it basically becomes invisible, right? And for this is the reason why generally you have to make a distinction. There's a rule of thumb: if you have a stained specimen, so basically one that has a natural color or that you have stained, then the refractive index of the specimen should be very similar to the surrounding mounting medium, right? Then, yeah, otherwise you've got diffraction happening, the light being bent, yeah, and then this kind of causes diffraction, and then you kind of losing the natural colors. So that's so-called, and the technical term is an amplitude specimen, because the stain, the color changes the amplitude of the light, the light, so basically it makes it darker, right? And then there are those specimens like those diatoms that are so-called phase specimens, and what they do is they change the phase shift of the light, and they should have a refractive index which is very different from the medium, so because then you want to use phase contrast to be able to see that, and you need phase shift for that. So this is getting a little bit technical, but maybe it just gives you a little bit of an idea that different mounting media are used for different specimens. And what I'm kind of doing here right now, a little bit the justification. I just want to show you with experimentation that indeed there are different mounting media that have different characteristics here. Oh, there's a question. Wow, really? That's a lot of questions, a lot of comments. Thank you. Okay. So, and last but not least, before I go through the questions, and then after the questions, I do want to show you some live specimens as well, because some people also want to see that. I'm going to do the following. You're going to now not believe it, but indeed, this has also been recommended once, okay, as a little try. Look, the craziest mounting medium ever. Yeah, it's not even, yeah, because somebody requested a hydrophobic. Cannot get more hydrophobic than that. That is immersion oil. Okay, so I'm going normally, so I'm not using the immersion oil now for its intended purpose. I'm just going to use it directly as a mounting medium, right? And let's suspend some of those diatoms in there as well. Okay. So my general recommendation is, because people keep on asking me which mounting medium to use, and I'm saying just use one that's easily available. Use some glue, yeah, at the beginning, because you just want to experiment around in most cases anyway, right? So that is now that's also very dense here. Maybe I also got a little too much here. So, and then on and the cover glass goes on top again. So that is immersion oil. It's synthetic immersion oil. In many microscopes that you probably got, you came with a bottle of immersion oil that had a yellow color. Yeah. Please, for those of you who are beginners here, do not confuse immersion oil with mounting medium. Please don't, okay? Yeah, mounting medium goes beneath the cover glass and the immersion oil goes on top of it. Yeah? So don't confuse those two. That's really, I'm talking from experience here because as a teacher in school, some students have confused this. And this can actually destroy the microscope objectives. Yeah. So that is not immersion oil. Okay, wow, it looks even darker. So let's have a look. Ah, okay, that's, I have to, that's immersion oil. Uh-huh. Again, a bit different. Yeah, this again looks different, interesting. There, you see that there's still some clusters in here. It doesn't seem to separate, yeah. Another thing that I need to say is the following. The mounting medium is not only a question of refractive index, but it has to be compatible with the specimen. If you actually see here that it kind of still, the diatoms kind of form clusters, right? This kind of is a little bit of an indication that the diatom shells are hydrophilic. So they don't like to be in contact with the hydrophobic oil and these tend to stick together better, which kind of makes sense a little bit because those diatoms actually used to live in water, right? So this cluster formation that you see here is actually like oil droplets in water. They also like to cluster together and make a droplet because they do not want to expose themselves to the water. And here, the diatom shells also don't seem to separate quite as nicely. They tend to cluster together because they want to avoid being in contact with the oil. Yeah. So this is actually another. It's actually quite. This effect is actually quite strong here. Yeah. The oil is still spreading because it did not cover the whole. Yeah. Let me refocus. Yeah, that's where there's no oil. Yeah. Can we not make this a little bit brighter? Yeah, let's turn up the light. Yeah. Yeah. So, for example, if you try the following, if you take a little piece of a bird's feather, and if you put it into water because you want to observe a bird's feather, then you're going to see that the individual parts, the barbules as they're called, they tend to stick together because the bird's feather is hydrophobic. So what you have to do is you just use just cooking oil, and this will kind of separate the feathers better, and there will also be less bubbles. Yeah. So yeah. So this is basically just, as you see, mounting medium is not only a refractive index issue, but also how compatible is it actually with the specimen. For example, you do not want to use Uprol, and I made a video some time ago, this Uprol mounting medium, very bad for example, for plants, right? Or because it really dissolves the chlorophyll. Right, the green pigment, yeah? Unless this is what you want to do, right? But essentially, some of these organic solvents will actually dissolve some of the pigments or even some of the stains that you've used. Yeah? So you see, it's there are many parameters, so to say, that you should consider. This one, for example, clearly clusters. Yeah, yeah. If you don't have immersion oil, you just try cooking oil. Yeah, but not for oil immersion microscopy. I mean, you just try cooking oil as a mounting medium. But if you actually want to do oil immersion microscopy, you need an oil immersion lens. That's really important. You basically, you put a drop of immersion oil then on the microscope slide. Generally, as I mentioned already so many times before, I'm not a big fan of oil immersion microscopy. Even though, because it's a messy thing, and you got to be careful. Are there actual subjects that Uprol might dissolve entirely? I've tried mounting Uprol already in very delicate water samples. For example, when there is a cell membrane, right? And not so much of a cell wall present in some microbes, then because there is a solvent there, it might actually dissolve the cell because the solvent will actually break apart the cell membrane, right? It dissolves the cell membrane and the cell flows out and then there's nothing left that you can see. Yeah? And that's why Uprol mounting medium is very liked for people who are doing insects. Yeah, arthropods and so on, because it does not dissolve the exoskeleton, but it actually clears it and brightens it. Yeah. Why did I choose UPROL mounting medium? I will be very honest. Not because I was so super interested in insects, but I chose it because it was for the following reasons. First of all, you can take a specimen from alcohol and transfer it directly into UPROL. That's possible. You cannot do that with other mounting media like UKIT, for example. This one, which I cannot try today because it already turned solid. So I have to add a solvent in here, xylene, which I don't want to inhale. So that's one of the things here. But UPROL is very suitable because you can actually take it from alcohol directly into UPROL. And therefore it's for practical purposes. There are not so many around, mounting media around, and that's one of the reasons why I was experimenting with so many others. That's why I was trying the nail polish, which is nitrocellulose, which has the disadvantage that it shrinks. Then luckily I found the Elmer's PVA glue, and now I'm also trying this glue over here, right, which seems to work in many cases also for water samples, because it's water-based, so it does not dissolve the pigments, right? Then I found for pollen you use the glycerin jelly a lot, because it has the right refractive index again, for making pollen visible. And if you, for example, I tried mounting UPROL with pollen, it destroyed the pollen, some of them, right? So you see, none of them is really ideal. Each one has its own pros and cons. Yeah. So I'm reading this here again, and I want to go through some of your questions. Paul is asking, is this mainly about refractive index, as diatom shells are basically glass? They are not affected by the mounting medium unless the medium contains a fluoric acid, I guess. Yeah, so this is the reason. That's correct. I mean, those diatoms are silica, right? And that's essentially glass. And those mounting media should actually not affect it. But again, especially for diatoms, you need the refractive index is super important. Yeah. What would it look like if one of the substances had lower refractive index? Would it be darker if the refractive index is too low and too similar to the refractive index of the diatoms, and the light is going to pass through unhindered, and you're not able to see anything? Yeah. So I go. Yeah. You know what? Let's just go to the questions first, and then I'm going to show you, just for the fun of it, a couple of water samples as well, because I've been collecting again some water samples. Again, I might have answered some of them already, so I'm going to quickly skip over those. Look, it's over 2 pages long. Thank you very much. Right, so I'm going to do this fairly quickly. Okay. I recently noticed a biofilm in one of the rocks. Great. Yeah, biofilms are always good to observe. How did you solve the USB problem? I bought myself a second USB card, and basically I've got now several empty USB ports. So in case you also are experiencing similar problems when you want to make videos. Infinite and finite microscope shares the same eyepieces. Yes. Why not? Because essentially the primary image is focused on the same place. Yeah, I would say it's not a question of finite or infinite. It's more a question also about eyepiece diameter. Yeah, if the barrel diameter is compatible. Yeah, I got bitten by a snake. Oh, okay, but you came back right on time to watch the live stream. I just started trialing with artist's watercolor drawing gum as a raised mount, and then seal cover glass before it evaporates. Drawing gum, I think they also used something called gum arabic, as it was called, and I think artists also used so-called gum arabic. However, I don't have a lot of experience with gum arabic. I assume it's gum arabic. Yeah. You also have to be very careful not to breathe in. Yeah, that's the silica. You don't want to get these silica shells into your lungs. Yeah. Yeah. We use this to provide chickens with an anti-bug floor. The roll in the stuff which you spray a bit around, it rids them of parasites when enclosed. Yes, because those silica are almost like nanoparticles, I guess. Yeah, and many insects don't like them. Yeah, it's an insect repellent. Here we go. Yeah. Yeah, is it a plant species? Well, officially it's not a plant because per definition plants are terrestrial. Yeah, but they're single-celled. Diatoms are single-celled and are related to the protists. Yeah. But of course, you can say that maybe they're also related to the plants because they do photosynthesis. Yeah, it's kind of like a category on their own. Staining these diatoms, you basically would stain the silica, but I can imagine that if it stains silica, then it must actually also stain the microscope slide. So I wonder how would that work. So I don't know. Yeah. The silica being inorganic, I don't know if some of the dyes will actually, the stains will actually react. Yeah. I mounted some diatoms in histological media refractive index of 1.5. They practically disappeared. I had to close the condenser almost completely to even see their outlines. Yes, this is actually 1.5 is actually very low. What I did some research for diatoms, you use 1.7 to 1.75 because 1.5 indeed is too low and then you do not see them very well. Yeah? So, and that's a little bit what I've been also trying to experiment around today with. Yeah. How do they collect these diatoms? Those diatoms in the diatomaceous earth are basically fossilized and they are simply mined. They're just basically found in the ground, right? I guess maybe also purified and so on. But those diatoms can be found in water samples as well, but these here are actually fossilized. Yeah? I assume that these fossilized diatoms are mostly broken. Under my microscope, they are often long and thin. This could be, indeed. Yeah, but I did find several ones that were also kind of more regular in shape. So I suppose that these were not so broken. But I admit that there are, I would say, especially marine diatoms, larger ones are quite nice and round and very huge and unlikely to find them completely intact. Yeah. Would a lower refractive index media work? Does it even exist? I don't know about that. Yeah, air. Air would be a lower refractive index. Yeah. Actually, I don't know. That would be something to try out. You just put some of, you know what, I'm going to try this out then, okay? Air has a lower refractive index and therefore, yeah, we can try that as well. Yeah? They often say fossilized. I argue that they are just old, not actually fossilized. There is no stone replaced. Okay, I get it. Yeah. Yes. Yeah. The term fossilized, you can debate that. Same thing also with, yeah, because your definition is basically stone replacing the shells. You could also say, you also say it's called fossil fuels, which is coal, gas, and oil, right? So the word fossilized or fossil, I think, might have different terms or different definitions. I get your point, yeah? What would polarized light or polarization filtering look like? I have polarized light now on my microscope. However, it's not regular polarization. I'd need a different microscope for that. And then you can actually, and what I think is the following, because it's just basically glass, I think they're just going to light up bright on a dark background, right? I think that's basically... yeah, I think the shells are simply going to depolarize the light, but I don't think that there are going to be any color changes. Yeah, so essentially it's probably going to be light on a dark background. Yeah, here we have. Yeah, so here we have a definition debate. Yeah, do you still use the large syringe? Yeah. White glue turning clear doesn't work. That's unfortunate. Yes, the white glue doesn't work. I also tried that. Yeah, the marketers and the manufacturers. I actually bought some Diatomaceous Earth. This stream couldn't have come at a better time, okay? It would be interesting to see if different brands of Diatomaceous Earth actually look different, yeah? I heard that when diatoms reproduce, they get smaller. Yes, it's like this, when a diatom shell, then the next one grows in here, right? And then they kind of become smaller and smaller. But then I think what they do, what I read somewhere, is they have a cycle of sexual reproduction in between where you basically are forming again cells that are big. Yeah. So they have this alternation of generations between sexual and asexual reproduction. And somehow this regenerates the original size again. Yeah. Blue nail polish, not tried. Well, let's put it this way. I can tell you it's so dark that you won't see anything. Yeah? So that's a little bit the problem. It's going to just make everything dark, and then you have the problem that you do not see any contrast to the specimen. Yeah? I've actually been, years, because we had cats that kept fighting fleas. Okay. Okay. Also for fleas. Okay, yoo-hoo, pronounced in England, is used for residential swimming pool filters. Available, say, okay, gardeners sometimes use small amounts with water to spray bottles to discourage aphid. Oh, that's an interesting one. We've got some aphid problems also in our garden. I might want to try this. Yeah. Yeah, you know what? I could try doing this. Not now because of lack of time, but I will take it on as a suggestion for a follow-up video. Thank you. Yeah. I tried the bright field. Okay, also in some hobby shops. I assume, honestly, I assume that it's probably going to be similar the composition as other water-based glues. Yeah, I assume. Yeah. Yes, I will taking pictures. I talked about this. I quickly go through here. UV, no, not tried so much. I know that this is one UV fingernail gel, which was too thick, actually, unfortunately. So that was the mounting medium. This was the immersion oil. Okay, I had to wait fully up to a week for the outer surface to fully cure. Yeah, it only absorbs water due to the capillary action with such a fine powder. Yeah, but this, yes. And then, of course, it would mean, and we've seen that it really absorbs water quite nicely. Yeah. You showed that you slide dry before washing, likely to kill microbes, but dried slides are hard to clean. What do you think about keeping them in soapy water instead? Okay, yes, it is correct. First of all, just because you dry the slides does not kill all microbes. You got to be still careful. There are some spores of bacteria that are able to survive even that. Okay, so yeah, I'm aware of this. And indeed, it becomes difficult to dry. What I bought myself is an ultrasound cleaner, and I have not tried it yet. But I want to try that. Indeed, I want to try to also heat fix some slides. And then see if the ultrasound cleaner, which is used for watches and jewelry and so on, if it's also able to remove whatever is kind of sticking to the slide, right? So this is something that I'm currently working on a little bit. I will report back on that. If you kind of rub the slide between your fingers when you wash it, you'll get off pretty much everything. But you don't always want to do that because, danger of injury and it's work and we get that. So what I, however, have found is the following: not soap, but use dishwashing powder. Because dishwashing powder contains enzymes and it will actually digest away whatever is on the slide. And dishwashing powder actually works quite effectively. Yeah. So let's move on quickly. Hydrophobic, interesting. Those are hydrophobic. Well, I think the diatoms are hydrophilic, or did I misspeak? Yeah, because they like to absorb water. I did some staining from your last stream with my daughter, and we had a great time. Oh, great! We even uploaded the photo to Instagram. Would it be okay if I tagged you? Sure, you can do that. Yeah, I used acetone-based clear nail polish to preserve an aphid larva. It worked great, and it also bleached as it cured. Oh, that's interesting. Okay, unfortunately, lots of air bubbles as it was too thick. And diluting it with more acetone stopped it fully curing in other tests. Yeah. So you see, it's kind of finding the right balance is difficult. What would it look like if one of the substances had lower RE? Yeah, that would basically mean it would be like in air. We can do a quick try on that. So how would it look like if we were to mount these directly in air? Very often, let's see, where is, here it is. You know what I'm going to do? So that should be enough. So this is a so-called a dry mounted slide. So let's have a look how this looks like. So where is the scope view? Oh yeah, this is again some. I have to find it. Maybe this one here. Huh. So you know what? I'm going to do the following. I'm going to just use bright field maybe. No, let's see. This kind of looks a little weird. What are these long things here? Could it be that when it's still in air, that it's kind of still clustered together? Yeah, they're still clustered. So that's basically how it looks like in air. Ah, maybe there's some individual ones over here. Let's see. Phase contrast. Here we go. But this again, they look kind of very small. Huh. Yeah, looks again different. You know what I'm gonna try just for the fun of it? I'm going to add a little bit of water. Just for the fun of it. Where did I put my water? And then, here it is, I got it. Let's see. Let capillary action do the rest. Ha! Ooh, this was too quick. They're all being flushed away, but it looks brighter now. Yep. Here we go. So that's now with water. So you see that, yeah, it does make a difference a little bit in how it looks like. Yeah. So I quickly scanned through some of your comments. Yeah. If there are any more, not so many. Yeah, you wanna see the pond water, okay? Yeah, I'll do that as well because you wanna see some live things as well. And is there any benefit to first placing diatoms on the cover slip, spreading the sample, and then covering it with glue to keep the sample close to the objective? Benefit to first placing it on the cover slip, spreading the sample. One could try that. Yeah, sometimes it is actually recommended that you actually put the sample first on the cover slip. But yeah, let's put it this way. I'll focus anyway. What you have to do is you have to find the focus in any case. Yeah. So I would say there are probably also other factors that play a role. And if you kind of press it really flat, then I think it should be in close contact with the cover slip in any case. Yeah? So let me quickly show you just something that I've got over here just a few days ago. I collected a water sample, and unfortunately I found out that it's a little too dark. Too far down. What, by the way, that's another project. I want to either use a laser cutter or a 3D printer, and I want to make some kind of a stand so that I'm able to put them in here diagonally so that it's easier to collect some samples. So let's grab a long pipette. So let's grab a long pipette here. Go all the way to the bottom. And take some out here. Rest goes back. And I know this is a lot, okay? I know this is way too much. And then what you do is the following. You just go like this. Okay. Rest can go back. At least before the live stream, when I tried this, I found a few nice things. And another thing that I want to say, I want to share a link to, I received an email. And with a request for sharing it. I will do that as well. I hope I don't forget. So, and I will do that while we're observing a few of these live things that we have in here. Scope view. Oh yeah, I have to remove this here. Let's go here. So I found a couple of even some paramecia in here. It was actually it's too small. So, and what we have here, of course, those long things that you see are of course diatoms. Yeah, and they are nice and green on the inside because they're chloroplasts present. And let me go up a little bit more. And then those long strings, I think they look like cyanobacteria as well. Yeah. And where is, yeah. I'm just trying to find some paramecia. Ooh, look at this guy here. This is looks like a gigantic crustacean. The shell of a this is a gigantic shell of a crustacean. Maybe it molted or died and then decomposed. And what you often see is that many of those cells are kind of clustered together. It's like also in a biofilm or in some kind of a polysaccharide matrix sticking together. Yeah. But those diatoms here are not really moving a lot. I have got a few other samples where they're more gliding. So you have different specimens that essentially do different things here, right? I consider them quite nice. Yeah. So what I'm going to do now is I'm going to find just a second. I need to find it first. There are two links that I would like to share because I received an email with a request of sharing those links because these are nice microscopy links as well. Yeah, also of how can I share them? I think I'm just going to copy them directly into the chat. Okay, that's basically, yeah. So this is a link 1 and link 2. And yeah, from YouTube channel Thai Microcosmos, and there are two nice documentaries, and one of them is about tardigrades and the other one is about diatoms as well. Really nice documentary style videos. And yeah, please have a look at them and subscribe them a little bit. Comment here, is there a way to dissolve that polysaccharide matrix? Yes, there is. What you do is the following. You can, and I have also a couple of months ago, I've done this and maybe I'm going to do it again. You take the algae with the diatoms and you treat it with hydrogen peroxide, H2O2, strong H2O2, 30%. And this is going to dissolve everything that's organic, and what's left over are just the diatom shells. And that's actually a way of extracting them. You can purify them. So you take a whole bunch of algae, and then you add the H2O to it, starts to bubble, you warm it up, and then you really digest it properly away, and then until you get a very fine white sand, like the diatomaceous earth that we've seen, and that contains then basically just the pure diatom shells. Looks very pretty. Because it's concentrated and all of the other things are removed, right? So the only thing is you got to be really careful. You got to be really careful with working with strong hydrogen peroxide because it can, of course, damage your eyes if you get it into your eyes, right? But that is actually what you do is if you want to take now that because where I'm living, it's springtime again, so algae growth starts again. And so what I can do is also in one of the upcoming videos, again, I can show this to you and how this is done. You take algae and then you add H2O2 to remove the organic material. Then you add acid to remove the calcium carbonate of any sea, how do you call this, shells and so on, snails and so on, so sand, which is calcium carbonate, and then you get more or less fairly pure diatom shells. Yeah, so which is quite nice. Yeah, yeah. Yeah, so what is this guy here? Yeah. If you want, we can look at it as at another one. Okay. And this one is the one from yesterday. And what I'm doing right now is this as well. In case you're interested, I'm making now a whole bunch of also unscripted videos. If you, yeah, please give me some feedback as well and which type of videos you want to see. I've been experimenting around a lot recently. I've got changed to the video formats and then some of the filming I did in my little in the cellar in my mini studio that I have, and then others I did basically unscripted. Yeah. I'm just preparing the next slide, unscripted here in front of the computer. So I've been experimenting around with a variety of different formats. And yeah, oh, you have not seen this now. What I've done is I've just taken the next water sample, and this one is from a different river. Yeah. And here you can actually see that the diatoms are again different. Yeah. Ah, yeah. Here the ones I've got those nice ones here that are going zigzaggy, and there are a couple one that actually start that glide and move around, which I kind of think is kind of nice, especially in time lapse. Yeah, if you do that, then you can actually see them. Yeah, glide. Yeah. Feeding an amoeba and see how big it gets. Yeah, I can tell you. I've been doing this as a matter of fact. Daniel is saying they're called slime molds. They can, they can become quite large. Yeah. So I'm just reading again the comments here. Yeah. Yep, I'm reading the comments. I hope I didn't forget anything here. Let's go next up. Yeah, I mean, really nice stuff here. Okay. So pond samples, feeding them with yeast. Yep. Would you be able to show us how to do it yourself in dark field for higher magnifications objectives? Ooh, dark field for higher magnification objectives is a problem. I will tell you why. Because it becomes too dark. Okay, what you have to do is you have to, if you want to make dark field filters, you have to make sure that the central disk for the higher magnification objectives, the central disk has to become larger. It has to do something with the numerical aperture. So basically then you have the following problem. The disk becoming larger means the whole image becomes darker. And in the high magnification, it is already dark, right? So what you have to do is you got to just make the disk larger, and then you can go up with the magnification. But then there's the problem that maybe you're not able to see anything at all because it's already too dark, right? So then you can only compensate this with photography and doing long exposure times. Yeah. So. Yeah, so that's basically just a common way to check your optics. If you let me turn on the arrow now, and then I think I'm gonna call it quits for today. Maybe not with live diatoms, but with and now you don't see it quite nicely, but with just diatom shells. What they have is they have those horizontal lines in them. I just was able to see them, but not anymore. And those horizontal lines, if you're able to see them with your microscope, then this is kind of like a resolution test. Yeah. So there are actually Diatom Slides out there being sold that are used for testing microscope objectives of whether you're able to see those structures. Yeah. Okay. Now the guy has gone vertical. So yeah. So just saying that there are also some how do you say testing mechanisms available that use diatoms. Yeah? And another thing I just want to show you here before I call it quits, and I also made a video on this, which I'm going to publish. And so look, just so that to make a little bit of sense of what we're able to see here. So that's of course an algae here. Yeah. So we're back in phase contrast. Yeah, and this algae, do you see those lines here on the surface? Those dark lines here, these are bacteria growing on the algae. Yeah. So this is essentially something that kind of illustrates a little bit of why phase contrast was also invented, because otherwise those bacteria are extremely difficult to see. So for example, when I remove the phase contrast again, yeah, yeah, they appear, you're still barely visible now, though, yeah. But with phase contrast, they appear dark, yeah, on a brighter background, right? And that is essentially the whole purpose of phase contrast microscopy. But in order to see this difference, you need the medium must have a different refractive index. Yeah? What is moving for diatoms? They have no cilia. Yes, they have a gliding mechanism, and I tried to research this, and I read that it's not fully understood how it works, but they seem to be secreting some kind of a slime on one side, and this kind of causes them to move. So it's a gliding motion, which is basically they adhere to a surface, and by secreting this mucus or the slime, it allows them to glide. But what I heard somewhere, it is not 100% fully understood of how this works. Yeah. Would you quickly get your diatom slide out to finish off? Did you find one? No, I don't now know which one you're talking about. Okay. No, I was just generally talking about that there are diatom slides. I don't have this one, but I've seen it documented in books and so on. These diatoms that are sold on certain slides, which are used for testing apparently microscope objectives. There are certain standards apparently, but I don't have that slide. Yeah. There is a comment here. Scientists aren't sure, but some believe that diatoms secrete some kind of mucus substance to move. Some research even compared diatoms' movement to our muscles. Okay. Maybe some kind of a sliding filament type of system. Yeah. Okay, so I think I'm going to do the following. I'm going to call it a day again, and maybe in one of the upcoming sessions, I'll see. I'll collect some more algae, and then maybe we're able to do a diatom extraction, okay, and hydrogen peroxide treatment to kind of purify, to purify those diatom shells. Yeah, so I think that's something that I'm going to do in the future. Yeah. So I'm going to. I hope I did not. I think I hope I haven't overlooked any questions. I think I'm just going to call it a day again. Day is good. Where I'm right now, it's almost it's 11 o'clock at night. Some of you also have very inconvenient times where you're joining. I would like to thank you for that, of course. And as Lawrence is just saying, I guess we just keep learning as we go on. And I also keep on learning a lot by all of this. And those slides I'm going to now dry, and we're going to have a look at them next week again. Okay, I want to wish you all the best. See you again next week. Happy microbe hunting as always. And I've got a couple of YouTube shorts and a couple of videos and so on. So plenty of stuff. Bye-bye. All the best.